Awesome. So I'm so delighted to have you all here for our first weekly Data for the People. So, as we mentioned, this is a new initiative where we're trying to share these journal articles in a way that the general community can understand. And so today I'm going to present our paper that came out that talks about how scientists can develop these tests that can test for COVID-19 immunity. Now, before I get started, I was given a brief intro, but I kind of want to give you a little bit more info about myself, just to highlight that even though I'm a scientist, I do have many parts of myself, and I am also human. And so I'm actually from the United States Virgin Islands, and I'm positive many people can't pick it out in the map, so I'm just going to zoom in. And we're actually part of the Caribbean, and so you probably still can't pick us out. But if you zoom in even more, we're southeast of Puerto Rico. So if you take a quick 15-minute plane ride, you'll hit upon the US Virgin Islands. And so I'm actually from the Big Island, and I say that in quotes because we're only 84 square miles, a population of 60,000 people. But I think the thing I want to point out today is that we all wish we were there. And so this is one of our lovely beaches on St. Croix. And as soon as this quarantine is over, we're probably going to find a flight to head home. But not only am I a Virgin Islander, I moved to the Northeast after college. So I did my undergrad at the University of the Virgin Islands. So we're HBCU in the Caribbean. We also had our own beach, which was amazing. In 2015, I got my PhD in microbiology immunology. So here I am with my mentor, Dr. Dana Mordew, who was very instrumental in that process. Outside of science, I love Beyonce. I love all things music. I guess you can say I'm part of the Beehive. Rihanna comes in a close second. But I also love all things science and all things fun. So here I am with Jeannie, our director at RockEDU, and we're just posing in really cool, funny lab gear in our yearly Science Saturday event that's catered for children in kindergarten through 8th grade. But not only that, but I do have a small family. So here's my husband and my son, Tyson. So this was in December. So we were also somewhere warm. So this was in St. Kitts, which is another Caribbean island where my parents are from at our family reunion. But he's now 8 months old. And I'm saying this because you might hear some noises in the background, and I'm going to try my best. I isolated myself in a room in the house that no one is distracted by the background noise. All right, so let's talk about this paper. So today I'm going to present this research on a paper called the Serological Assay to Detect SARS-CoV-2 Seroconversion in Humans. And there are a lot of fancy words in here. I just want to point out what they all mean. So serological is pretty much talking about the serum, which holds the antibodies in humans. Assay is just another word for tests. And SARS-CoV-2 is the virus that causes the COVID-19 disease, which I'll talk about in a few. And seroconversion is just the transition from being infected to producing antibodies against a specific infection. And we're all human. So in other words, all this is saying is that this paper looks at developing a test to see if people who have been infected or who have recovered from this coronavirus disease. And so we pointed out a little bit earlier, talking about like science is collaborative. It encompasses many scientists who are working towards a similar goal. This one paper in particular have not one, not two, but 19 authors. And this is to show that this is not a one-man show. And these authors are listed in a specific order, which highlights their role in this paper. So for example, I highlighted Fatima Aminat, and Fatima is listed as the first author, which usually means she's the person that kind of did the bulk of the experiments and the analysis. All these other authors were instrumental in producing the data that they have, the results, and some of the other analyses. And then the last author, which is Florian Kramer, is usually the person that funds the project or oversees that project. And so like we also mentioned, not only do we have many persons working on this specific science experiment, but they're coming from all across the globe. And so this particular paper encompasses scientists from New York, which is in our background. We also have scientists in Melbourne, Australia, in Finland, and in Switzerland. So I kind of like to think of science as a universal language, but also it's really cool to see that someone can perform experiments in New York and can reproduce those same experiments in Finland and usually get similar results. So we've thrown this word out, scientific literature. But what does that mean? So scientists usually put their data in a package, report it to the general public. And so these reports describe the latest research that's been happening from across the world, but then these literature have to be vetted by peers. And so our knowledge is shared within the scientific community. We never want to work on a project and not share those amazing results or those techniques that led to those results. But the problem is with a lot of the scientific literature, they're usually published in journals, and these journals sometimes require a subscription, which is usually expensive or costs some amount of money. And if you're not affiliated with a university, it's really hard to kind of get those articles. But also, a lot of the journal articles that are on there use very specific jargon that makes it difficult for not only non-scientists, but non-specialists in the field to digest. So for example, my background is microbiology, immunology, and it'll probably be really difficult for me to read a paper that's written by or that surrounds a project in astronomy or astrophysics. And so the thing we want to highlight though that in these scientific literature, there's knowledge in there that directly impact our lives. It's the basis for the new technologies that we see, the healthcare options that are given to us, but it also deepens our knowledge on our own biology. So there are two different ways that people can access these papers. And so one is what we call a preprint, and the other is what we call a peer review. So what's a preprint? So usually scientists can come up with their work, put it in a really nice way to put it on paper so that others in that community can read. Now, this is a really nice way that scientists are showing this data in real time. And so they're uploaded to a specific server, and people in the community, including scientists, can actually read that paper and can share feedback directly to the authors on the paper. So the value is that you can get it in a timely manner. You can receive the feedback from your peers, but also you can, it's a really cool way to review what's happening in that moment. And so, but peer review is another way that scientists can share these journal articles. And so this is a process in which these reports are actually go through a process where it's vetted by experts in the field. So say these scientists decided that they wanted to put this paper in a specific journal or organization. That organization would recruit scientists that are experts in that field. They read through the data, they give comments back to the authors. They say, hey, this is a great paper, but can you please add on these couple experiments? Or, hey, I think if you added this extra stuff, it'll be really good, but at this time, we're not going to accept your paper. Now, this is a gold standard for ensuring quality and rigor, but this process can take several months up to years to complete. So in a time like now, it's really cool that we have this preprint where we can actually see the data and the experiments that are popping out in a timely manner. And so this paper came from one of those preprint servers called MedArchive. And so this is just a preprint for papers in the health sciences, but there's also BioArchive, which is a server for biological sciences. But I just also want to note, because it's not peer reviewed, they also caution that these are preliminary reports of work that has not been peer reviewed, and we should not rely on it for any clinical practices, health behaviors, or should report in news media as established information. And so this is where this paper came from, MedArchive. So I want to talk about the styles of scientific reasoning. So there are many ways to attack a question in science, and we can approach it in many different ways. Now, the one way that this paper approaches a specific question or a goal is through a style that we call experimental evaluation. And so this means that the study involves many rounds of repeating those experiments and documenting those results, and it's what we call a controlled experiment. So let's go back to when you were in middle school or high school. Most people have done a similar type of experiment. And so, for example, people who enjoy growing plants, right? And you have a question, would my plant grow better in yellow light or red light? And so all of the conditions are the same. You use the same plant, you water them the same, they have the same nutrients. The only variable or the thing that's different is the type of light that is shown on this plant. So we have a plant under yellow light and we have a plant under red light. You let some time pass, the same amount of time for both ways to assess the plant growth. And after that time, you look and see what those results were. And so in one case, you'll see that, hey, my plant didn't grow as much under the yellow light, but it grew a lot under the red light, and I kept everything else the same. So I controlled my environment. So this might hint to maybe red light has something to do with plant growth. And so again, this is what we call controlled experiments, and this style is known as experimental evaluation. So before we get started on the paper, I want to do a poll. So I want to get everyone involved. How big do you think a viral particle is? All right, so we'll put up a poll shortly that everyone can answer. So here we have the question. So how many individual virus particles do you think can fit on the tip of a sharpened pencil? Is it 500? Is it 5,000, 5,000,000, or 500 million? So I'll give you a few seconds for everyone to vote, and then we'll share those results with you. Okay, so we'll end the poll in about 10 seconds. Awesome, so let's share those results. Okay, so 52% of you who voted actually voted 500 million. About another half, 44%, voted 5 million, and a subset of you all voted 5,000. So the correct answer is 500 million. Imagine that 500 million virus particles can fit on the tippy tip of your sharpened pencils. All right, but let's visualize that. It's kind of hard to like see it, but I want to show you how tiny these viruses are. So I'm just going to go to this link, which is super fascinating to see from in Utah, University of Utah. And it scales the size of things that we know about, um, and shows how small viruses are. All right, so many of us have had that cup of coffee this morning. So we're starting off with a small coffee bean, and we're zooming in, and we see a grain of salt. We start zooming into the unicellular organisms like an amoeba and a paramecium. Now we have a human egg. We can now visualize as one skin cell. We have that tiny sperm going towards the egg, a single baker's yeast, the X chromosome. We still haven't reached viruses. Now we're at a single bacterium, E. coli, the powerhouse of the cell, the mitochondria, the lysozyme, which degrades compartments in the cell. And finally, we're at these viruses. And these viruses are super tiny, but their sizes also range with the type of viruses that they are. And if we zoom in even further, you'll see some of the cell's machinery in there. And one thing I also am going to talk about a lot is this antibody, which is also really tiny. So now let's talk about like the main topic at hand, coronavirus. All right, so what is coronavirus? Like what are these terminologies that's been used in the media? And so COVID-19 is the terminology that we use to describe the virus, the disease itself. So CO, the first two letters in corona, VI, the first two letters in virus, D is the first letter in disease, and the first confirmed case in 2019. Now, this virus, COVID-19, is caused by a specific type of coronavirus. And we've been calling it the novel coronavirus, but it's officially called the SARS-CoV-2. Now, SARS stands for Severe Acute Respiratory Syndrome. And for those of you who remember, in 2002, we had a previous SARS outbreak. And this particular coronavirus is actually closely related to this SARS-CoV-1. So we've given the name SARS-CoV-2 for this novel coronavirus. Now, the name coronavirus may be new to many people, but these coronaviruses have been around for a very long time. If you've had the common cold, chances are that might have been caused by a human coronavirus. And so there are many different types, but this particular one is a beta coronavirus, and we named that SARS-CoV-2, or scientists named that SARS-CoV-2. And here's a really pretty picture I think has been circulating many places. And you can see that these proteins or things on their surface kind of look like crown. So corona is Latin for crown, and so hence the name corona. But how does this virus get into the cell? All right, and so how do we get sick? So here's an image of the virus, and I want to spend a couple seconds focusing on what this virus looks like. And so viruses encompasses their genetic material inside of its particle. In our case for SARS-CoV-2, it has RNA. Unlike humans, where our baseline genetic material is DNA. And so this RNA is protected by a layer that we call the lipid bilayer, but in other words, it's just a layer of fat. And that layer of fat has a number of different proteins on the surface. Now, these proteins, this one in particular, in which we call spike protein, is very important for a couple things. One thing is that it's important for getting into the human cell. So in order for this virus, the coronavirus, to get into the cell, it has to be a really good match to the proteins on our human cells. And this particular protein in this case is called the human ACE2 receptor. So it's kind of like a firm handshake. So the virus is disguising itself like, hey, you remember me, let's shake hands really firmly. And once it shakes your hand, it puts itself inside of the cell. And so the virus needs to be inside of a whole cell in order to make more copies of itself. And this is why scientists has come to a general consensus of viruses not being alive, because it can't make more viruses without the help of a whole cell. So these whole cells provide the machinery needed to make many more of these viral particles, which can go on to infect more cells. But this spike protein also have another role. And it has to do with the immune response. And so by now, many of us have heard oh, do we have antibodies for the virus, right? And how do we know if someone is infected by the virus, and will we be protected? And so I want to briefly talk about our immune response to this virus in particular. So our immune response has two players. We have the antibodies and we have the antigens. So what the antibodies are these Y-shaped molecules or proteins that are produced by our immune cells, so say our white blood cells. These antibodies recognizes foreign particles, and in our case, those foreign particles would be coronavirus, the SARS-CoV-2. Now, the antibodies realize that, hey, this is something I don't recognize. Let's find it. Let's bind to it. Let's attack it, get rid of it. But not only that, let's remember this virus so that the next time it comes around, we can quickly clear the infection without that human getting sick. And so the thing that these antibodies bind is what we also call the antigens. So it's a dual play role. So you have the antibodies, which bind to the antigen, which can, one, get rid of that viral particle, and two, it can send messages to other immune cells in your body to remember this particular antigen, so the next time it comes around, we can quickly get rid of it. And so these two terms are very important for this paper. Not only are they important, but we actually have many different types of antibodies, 5 to be exact, 5 main types. But the two I want to focus on are IgM and IgG. Now, IgM is usually the first antibody to respond to a viral infection. And IgM is usually swims around in a pentamer, which just means 5 molecules put together. So it's like a 5-molecule complex. And the second antibody that comes into play is the IgG. So the IgM kind of sends messages to your body and your immune cells like, hey, I'm giving you this information to make sure that you can capture this the next time it comes around. So IgG is what we call the secondary response to this infection. So let's talk about the testing for COVID-19. And so right now we have tests that can tell if someone has that virus. And so you guys have seen where there are videos floating around where they take a swab and they stick it down their nose and they see if someone has that infection. What we lack currently are tests to see if someone has already had the infection. So on this scale, we have the number of days and we have the amount of that particular thing that's present. So someone gets infected with that virus. Oops. And what happens is that viral load increases, kind of hinting to the immune system, hey, something's going wrong. And then we have this IgM antibody coming in that starts attacking this virus, but it's also sending messages to your other immune system. And now your IgG antibodies come into play, which is your secondary response, and it increases over time. And we usually call these people immune to the infection because once you have these IgG present, as soon as you might get exposed to this virus, it can quickly attack it. But how do we know that people are protected from this virus or that they have produced antibodies for this infection? So this is where this paper comes in. So one thing scientists ask is, what information do we have so far? What's lacking? What do we need to work on? So just to know, this paper came out about a month ago on March 18. And at that point, we have and still do have tests or molecular assays. So it's basically a test that looks for whether or not the virus is present. So we consider that a current infection, but we don't know when that person clears that infection fully. And we also don't know when they've produced these antibodies that can protect them the next time around. And so this paper really focuses on, like, can we develop a test to see if someone has been previously infected with SARS-CoV-2? So what's the main goal of this paper? So once again, they want to see if they can develop a version of this virus, and they're going to use a particular protein of the virus that can act as that antigen that I talked about, and see if we can use that antigen to detect whether or not someone who's been infected do have the antibodies in their blood. So once again, to reiterate, our bodies are really good at pumping out these antibodies when we get sick. In our case, we're going to talk about SARS-CoV-2. And again, these antibodies specifically recognize the proteins on the surface, which we're going to call the antigens. So this antibody-antigen complex or interaction is very important for the rest of this paper. So again, we have this virus. And the thing with working with this virus is that it's very contagious. You're putting people at risk and at harm. And so it's really hard for scientists to take whole viruses and try to use it to manipulate to come up with tests and things that may help with combating this current pandemic. So is there a way that we can use part of this virus that can recognize the antibodies in the immune system without causing harm to the scientists or when we're formulating these tests? So what these scientists did is that they came up with a way to make this particular protein, which is called a spike protein, in a test tube and use it to figure out whether or not we can use this one protein and not the virus to detect if someone do have these antibodies. But remember, I told you these proteins or these viruses are really small, antibodies are really small. So scientists have a really neat way to actually visualize these proteins. And we call this a gel, right, for the lack of a better term. And with this gel, you see a lot of blue markings. So these blue markings are all proteins of different sizes that we use as a ladder or a control. Now, scientists know by coming up with the methods for this protein, the approximate size of this protein. So here we see the different sizes, and KDA is kilodaltons, and it's just a way that we measure proteins. And so they know the approximate size of the proteins, but in this particular gel, all proteins that they're able to produce in that test tube would light up this blue color. So what you can see, the scientists are confirming that they were able to synthetically make in a test tube just that one piece of protein from the virus. But now we have the virus, which we'll call the viral antigen actually, and we have this protein that's going to act as that. And so what we want to figure out is, okay, we don't have the virus anymore. We're not at risk, but can we take that small synthetic viral antigen and use it to see if someone has been previously infected with the SARS-CoV-2? So just to remind everyone, when someone gets infected with coronavirus, the antibodies bind specifically to the antigen. In our case, that viral antigen is now that spike protein. And we want to see if your body's antibodies to that infection can recognize just that small part of the virus. So they developed a method, not the method itself, but the way that they developed it is fairly new because they're using new molecules or new components. But this method has been out for many years. It's called an ELISA, an enzyme-linked immunosorbent assay. So how many of you guys have seen what a pregnancy test looks like, right? Or like the HIV over-the-counter test. They all utilize this type of methodology to see whether or not something is present or something is absent. Now, remember, these proteins, our antibodies are really small, so the enzyme linked is a really nice way for us to actually visualize if that thing is present. So in the case of this paper, we've developed this viral antigen in a test tube, right? And we have a number of different samples from humans who've been previously infected with coronavirus. Now, the thought process is that in their serum, which holds their antibodies, if their serum are SARS-CoV-2 specific, the scientists are hoping that it can recognize piece of that virus. So in the test tube, they add the serum from the human that's been infected with coronavirus. And if they have antibodies that can bind that viral antigen, it does so. But how can we see that it actually is binding to that viral antigen? We have these synthetic antibodies that have this really cool link on it that can actually allow us to visualize it in a test tube, right? So after we have that binding taking place, we add an activator. And so it goes from us not being able to see anything, right? So if there's nothing being shown on it, we're going to assume there's no antibodies present. But if something is there that's binding to that viral antigen, you'll see a color change, right? So basically in this assay, what scientists are looking for is whether or not this antibody is present, so color is there, or if that antibody is absent, there's no color there. So what does this look like when we graph it? So what they were able to detect is that the antibodies from people who have been previously infected were able to detect that small piece of that virus that they made in the lab, which is the viral antigen. And so I just want to hone in on the axes. And so on the y-axis, you have what we call the optical density, or basically the amount of light that you can see through a machine. And so the higher that number, the more antibodies that patient serum have that bound to that antigen. The lower the number, the less antibodies that they have towards that can bind that antigen. But we also have background signal, and so scientists control for that amount. Now on the bottom, we want to ensure that it is that antibody that's causing that light signal. So one thing scientists can do is actually make dilutions of that antibody so that we can make sure that it is the antibodies that are binding to that antigen. So what they do is kind of like having a really sweet juice. Like, so we do this all the time. I'll have like the Welch's passion fruit juice, but it's really sweet. So I dilute it like half and half with water. So every dilution, I'm adding more water to it. And so you'll get to a point where it's super diluted and you don't see a signal because there's very little antibody in that test tube. Now we're looking at the different serum samples that they're using. And scientists did a really neat job of adding in these controls that I talked about in the beginning. And so we have we actually have 4 serum samples that came from 3 persons who were infected with SARS-CoV-2. And so here we have patient 1, 2, and 3. So 1 and 2 are the red and dark red color. And you can see there's really high signal leading to the thought that these people who've been infected with SARS coronavirus do have antibodies that can recognize the antigen that these scientists made in the lab. But then we also have this other patient. So we have at 2 different time points post-infection. And you can still see that they're able to read out a signal ensuring that the antibodies from this virus infection is showing up and is binding to the viral antigen. Now, what are the other controls? So they had about 50 other patients that they got serum from in like 2019 before the pandemic reached the US. And so these control samples, we can say that these control samples have actually been humans who've been infected with other viral infections. So they've either been infected with the flu, they've probably been infected with other coronaviruses that I talked about earlier. And so we want to ensure that this test and this antigen is not actually binding to antibodies that should be specific to SARS-CoV-2. Now, this one I think is really neat. So they added a sample of someone who had a confirmed case of another human coronavirus, and that's called NL63. And this human coronavirus is kind of special because it actually uses that same human receptor that SARS-CoV-2 have. So they may have some similarities in that antigen or on that protein on their surface. But you can see that this assay is really specific because the antigen that it produced, the antibodies that may have been in that serum, does not recognize an antigen that comes from the SARS-CoV-2 virus. All right, so I've shown you that the antibodies in the serum can detect this antigen that they've made in the lab. All right, so just to reiterate, we have the virus that infects humans, and then we have these other data points where you see a production of IgM and IgG. What I did not show you was this test that I showed you previously, or the results, did not indicate what type of antibodies they were. All we know that there were antibodies in there and they were specific enough to bind to that viral antigen that they made in the lab. So how can we see whether or not it's IgM or IgG or some other antibody that's binding to this viral antigen? So what they did is something that we call isotyping. So it's basically just trying to figure out which type of antibody is actually binding to our, viral antigen. And so in this slide, I'm showing one data point where they test and see the different types of antibodies that were present. And so the two that I wanted to highlight were IgG, so IgG is present in 4 different subtypes. And then we have the IgM. Just want to zone in on the scale. So these are 10-fold marks. So this is like a log scale. And so the amount here is 10 times higher than the amount below. So it shows us how much of that is present. And the x-axis just shows us the type of antibodies. And so all of the patients, you can see with the different differentiations, is that they all can, they're able to detect IgG present and IgM present. But one thing that's been going around in the media, so if you guys watch CNN and you listen to Cuomo or any other news outlet, you hear them talk about like, we need to make sure that these antibody tests, when we want to bring them out, are really good and not going to give us any bad information. So one thing that these scientists want to make sure is that this test is both specific and sensitive. So what do I mean by that? So specificity. And so we want to make sure that if someone else has been infected from another viral infection, if they were to take their test, it's not going to come up saying that they have antibodies to it when it's not the actual virus infection that we're looking for. So in this case, what these scientists did, remember I told you that they also tested someone with a confirmed human coronavirus case that bound to the same receptor, but this test was so specific that it did not show any light, meaning that the antibodies in this patient's serum did not bind to the viral antigen that came from the SARS-CoV-2. Once we can ensure that this test is specific, we can then reduce the amount of false positives. So we don't want the test coming up positive when in actuality you had a human coronavirus. You go out into the population and thinking that you're immune, but in actuality, it was just another coronavirus that you had. Now, the authors also try to talk about like sensitivity. So sensitivity meaning like how soon can we detect if this person was able to produce antibodies to this infection? So if this person got infected, say 2 days ago, is this test sensitive enough to pick up on maybe the little bit of IgM that's being produced? And so I just highlighted one of these serum samples. This person was infected about the serum was taken from this patient about day 4 after infection. And you can see that there is IgM present that they were able to detect, and there's also IgG present. And so it does seem to be able to pick up antibodies that may be coming in early in infection. And so once your test is super sensitive, you actually reduce the amount of false negatives that you have. So you also don't want someone going out and be like, oh, the test came out negative, I didn't get infected with coronavirus, and then, go out and is susceptible to the thing, and, maybe they were already infected with SARS-CoV-2. But this paper, I only show you some of the main data points, but this paper actually did a number of different things. And I kind of want to zone in on the process of science. Like this didn't come about in like a day or overnight, right? It took some time, it took some trial and error. And so these scientists actually try to produce this viral antigen, so part of that virus, in different types of cells. right? So different types of cells, we can hijack the machinery in those cells to kind of make the protein of interest. And I did not show that data. The scientists also try to make different versions of this antigen, right? So not only could you make the full spike protein, maybe you just make the small tip of it and see whether or not that works. And so I didn't show that, but the spike protein actually worked better in their assays or their tests, other than the other versions that they tried to make. I mentioned that in this study, they only tested 4 samples from 3 patients. So I also want to point out that this paper was just to develop the assay, and it was not a clinical trial. So what scientists can do, they can take this information that was on this paper and use it in a clinical trial where you're testing a larger number of humans to see if this test does actually recognize on a broader scale. And what I really love in this paper is that they make this point, and this is what I really like about MedArchive, even though it has not gone through the peer review process, is that we're all working towards a similar goal. We're all really trying to get over this pandemic as soon as possible and go back to what used to be normal, right? And so at the end of the paper, they made sure that they stated, we are making all the methods and reagents widely available to the research community in order to support this global effort to limit and mitigate the spread of SARS-CoV-2. And I just know a lot of people think that, we like work in a laboratory, we don't want anyone else to have this information. But in reality, this is such a collaborative effort that we're pumping out this data and we're putting out the papers on there so that other scientists can go into their labs, utilize the methods that they have, and see if they can even perfect that. So what does this look like in the future? So I spoke about their testing that are available, but none of them are FDA approved yet. And so I actually found there are 2 companies' websites that actually already have this antibody test. All right, so this is what it looks like. If anyone has taken a pregnancy test before, it looks very similar to this. And so on the test strip, you have a control line. And so having a control, you're ensuring that the test works. So you're making sure that there's something that's consistent in everyone's blood or serum. So this test is actually a prick on your finger. So you prick your finger and you put the blood in the test. And so if this light comes up, you know that the test works. Now you can also have a result that's IgM positive. So remember I said that IgM is that antibody that responds first to this viral infection, right? And usually if someone is IgM positive, that means that they're probably still fighting off that infection. So this person is starting to produce those antibodies, but still have the virus present in their bodies. So you shouldn't leave home, right? You shouldn't leave home either way, unless you're an essential employee. But we also have a test that could be IgG positive. And so what IgG positive, remember I mentioned that this IgG is the second one to come into play. And usually at that point, you've already cleared the infection and you can make the assumption that you are immune to infection, so you're not susceptible. And so these are the patients that we're talking about in the news outlet, especially for healthcare professionals, like, okay, you've recovered from this virus, you have the IgG, you're immune. But not only that, a lot of people who have a lot of IgG present, we can actually utilize their serum to help save the lives of people who are trying to recover from this illness. And then you can be in a phase where you're IgM positive and IgG positive. And this just means that we're not too sure whether or not you've cleared the virus completely because 2 antibodies are present. And one thing I also want to point out is that the site also mentions that this test is not necessarily as specific as it should be. So they state that these positive results may be due to past and present infection with another coronavirus strain that I've talked about previously. And the one that they mentioned that I want to highlight is that NL63, which is a human coronavirus that also binds to the same human receptor. So this is why I think the FDA is holding out on approving some of these tests, because you don't want people thinking that they are immune to it, go out, get the disease, and can spread that to someone else. So moving forward, at this point, we have the testing available to see if people have current infection with the virus. Granted, there's all this debate and argument about like making that accessible to everyone, but also pointing out that, remember, even if you don't have symptoms, that doesn't mean that you're not infected with the virus. And I think this is why social distancing is really important with the test that we currently have, because we have no way to test if someone has developed immunity to this current infection. And I think scientists are working really hard, and it's not a matter of like, if we're going to have that test, but I think it's like, when is that test going to be sensitive and specific enough so that the general population can test and see whether or not they've already recovered from that virus and they're protected for some amount of time. So that's all I have. Thank you. And I'd be more than happy to take on any questions.